Hi, everyone. Thank you for coming to MIA today. I'm really excited to present two amazing speakers who've come from Hopkins to join us. First, we'll have Caleb Hallinan. He's a third-year BME PhD candidate at Hopkins, and his research focuses mainly on predicting spatial gene expression from histology images using neural networks. However, today he will be introducing spatial transcriptomics and present recent work on probe binding issues using 10x genomics Xenium technology. So if we could give him a round of applause and welcome him.
Great. Thank you. So hi, everyone. Again, my name is Caleb Hallinan. I'm a third-year BME PhD student and Dr. Gene Fan's lab at Johns Hopkins University. And today I'll be talking about spatial transcriptomics and then a recent project I've been working on where we evaluated probe accuracy and the 10x genomics Xenium platform. So what is spatial transcriptomics? That's the question I want to answer to start. I feel like this is a big word, and I tell my friends and family, and they have like no idea what I'm even saying when I say this word. So how I'm going to explain it is with this analogy. So there's a couple ways we can measure gene expression today. So say we have an organ, and in this case, our organ is a fruit basket. So one way we can measure gene expression is bulk RNA-seq. So bulk RNA-seq in this example is like I'm taking my organ, my fruit basket, I'm putting it in this blender, and out comes a smoothie. So in this representation, we have tissue-level gene expression. We have no single cell or spatial, just tissue-level gene expression. So next, we have single-cell RNA-seq. So single-cell RNA-seq is we no longer have tissue-level expression. We can have single-cell level expression. So in this example, we have our organ, which is the fruit basket. And with this fruit basket, we have a bunch of fruit. And so we can get exactly what fruits are where and what fruits are expressing genes. And yes. And then we have spatial RNA-seq. And so spatial RNA-seq is, say we have this fruit basket and it becomes a fruit platter. So we know exactly where these fruits are exactly in this fruit platter. We know where the strawberries, blueberries are, et cetera. So with spatial RNA-seq, we can do the same thing. We know where genes are expressed spatially in this tissue. So the one sentence definition I'll give for spatial transcriptomics is it measures gene expression in intact tissue while preserving spatial coordinates. So that's kind of the overview of spatial transcriptomics or what it is. But today I want to talk about how do we get from the organ to the spatial RNA-seq? How do we get from this fruit basket to this spatial platter? So from a data analysis, I would say there are two main types of technologies we'll focus on. The first one is an imaging-based technology. So the example I'll use is 10x genomics Xenium. So I'll explain this in more detail later on, but the way 10x Xenium works is you're basically sending in these probes, and these probes will bind to mRNA transcripts, and then you take a picture and you can see where that mRNA is in the tissue. So what's cool about this is it's single molecular resolution, so we know exactly where mRNA molecules are present and where they are. It is a targeted gene panel, so we can get like 5,000 to 6,000 genes at this point with this technology. It has a high detection efficiency. So it's really good at finding the target genes that you're interested in. And again, this is probe-based, so imaging-based technology. So what this actually looks like is, say we have the single cell, and we segment the cell so we know the boundary and the nucleus, where the nucleus is. So within this cell, there's a bunch of mRNA molecules present throughout. So with these technologies, we know exactly what gene it represents this mRNA molecule. So we know these are gene A, these are gene B, and we know for up to hundreds to thousands of gene species where these are present in the single cell. And so if we can do this for a single cell, we can do this for many cells. So here we have many cells around each other. They each have mRNA molecules present within. We can segment them. And if we can do this with many cells, we can do this for an entire tissue. So here I'm showing a mouse brain, and this is a 10x Xenium results. And each point represents a single cell in this data set. And so what this looks like for us as computational biologists is this matrix, where we have on our rows the genes for this mouse brain, and on the columns we have our cells. And so we can get, again, tons of genes and tons of cells, and we get this massive matrix. And so the way that we typically will visualize this data is if we plot a gene, let's say gene RASGRF2, you can see that it's pretty highly expressed. Let's see, up at the top here, and then these middle areas. But it's kind of hard to see. It becomes a little bit difficult because, again, each of these points are single cells, and we're plotting the gene expression for this gene for these single cells. So one way we can visualize this gene a little bit easier for this technology is by rasterizing it. So what we do is we aggregate all mRNA molecules to patches. So each of these patches, I'm taking all the mRNA molecules within this patch, I'm summing it together. And so now when we plot this data, we can plot it like this. and it's a lot easier to kind of visualize the trends in these genes and what these genes actually look like. So here's the example of RASGRF2. We can do this for all the genes, FezF2 or RORB, et cetera. And so another technology that I'll talk about is 10x Genomics Visium. So this is a yeah.
One question is, other than the visualization purpose, what is the benefit of putting them into the grid?
That's a great question. I think I'll answer it briefly. I think there's a computational benefit. So we're reducing the single cell, which is like a ton of data, up to these patches, which reduces the data. So it's a little easier computationally down the line to run things. It won't take as long. But hopefully, Dr. Fan will be talking about that a lot more in her talk. And so she'll give you a better answer of why we do it and other reasons. Yeah. Cool. So again, the other technology we have is 10x Genomics Visium. This is a sequencing base. So the way that these technologies works is they have spot and pixel resolution. So instead of singular molecular resolution, we know exactly where the mRNA molecule is. Spot and pixel resolution is we know, in this case, our example is 10x Visium. Within this 55 by 55 micron spot, we know what mRNA molecules are present within there. So again, no longer single molecular, it's more of a spot pixel resolution. What's cool about this technology is we can get full transcriptome. So full transcriptome, we're getting all 20,000-ish protein coding genes compared to our imaging base, which is 5,000 to 6,000. However, this does suffer from a little lower detection efficiency. So we're not able to get as high detection efficiency compared to imaging-based technologies. And again, this is sequencing. So we're actually sequencing the mRNA molecules that are within this spot. So if we were to actually look at what this might look like in an example, say, again, we have these many cells. In this case, we don't need the cell segmentation because, again, all of these mRNA molecules will be aggregated within this 55 by 55 micron spot. And so if we can do this for many cells, we can do this for many cells within many spots. And so we can do this many cells, many spots. We can, again, get a tissue of this data. And so this is a mouse brain, half of a mouse brain with 10x Visium cytosist. And we can plot those same genes. So this is RASGRF2, FezF2, and RORB. And hopefully you can appreciate that they have very similar gene expression, detected gene expression, whether it was the sequencing-based or imaging-based technologies. So this is spatial transcriptomics. Again, it's measure gene expression in intact tissue while preserving spatial coordinates. There are a lot of downstream analysis that we can do with spatial transcriptomics. Dr. Fan will be talking a lot about what we've been doing in the lab to analyze this type of data. For me, I'll focus the next part of this talk on the differences between these two technologies, or really focusing on the Xenium technology. So when we're using these technologies, they are doing different things of like collecting the data, but ultimately they're collecting the same type of data. We're collecting gene expression, spatial gene expression. And so like this example, RASGRF2, we would expect them to be the same approximately if we were to plot the genes, which here you can see it is. RASGRF2 seems to have the same spatial profile regardless of whether you use the sequencing base or imaging base. However, when we were looking at these two data sets, data set, we found that there were actually some differences between these two technologies that we weren't sure why. We didn't quite understand what was going on. And this is the data set that I was looking at when I found this issue. This is a data set from a previous publication from 10x Genomics. They had a breast cancer tissue block, and they took serial sections of our tissue block. One of these serial sections went to Xenium, which is, again, the imaging-based platform. Another serial section went to Visium, which is the sequencing-based platform. So we took this data from their website, and we did a little pre-processing where we aligned the data to the same tissue, and we kept it to the same shared areas. So you can see here, this is our shared area between the Visium and sequencing data. And then we aggregated the Xenium data to patches, as I mentioned earlier. So that way, both of these data sets are on the same molecular resolution. So they're both 55 by 55 microns. Here, the Visium, I'm just plotting it in patches instead of circles. So now it becomes really easy to compare these two technologies because, again, these data sets are aligned. So at each patch, they should have approximately the same gene expression, regardless of what technology I use. So when you take a look at a couple of these genes, like MS4A1, we see that this is the Xenium expression of MS4A1, and this is the Visium expression of MS4A1. You can see they're pretty similar. The spatial pattern is relatively similar. The gene expression is a little different, but again, imaging typically has a little bit higher detection efficiency, but overall, very similar. And we can do this correlation analysis and see that it has a pretty moderate agreement between, this is patch level correlation. You can see our Pearson R is 0.382 and our RMSE is relatively low at 3.746. So I'd say this is a good example of a gene. However, we found genes like this gene, ApoBec3b, where in the Xenium data set, you can see it has this really cool spatial pattern. It's pretty well expressed. But in the Visium data set for ApoBec3b, there was no detected expression of ApoBec3b. And so if there's no detected expression, obviously there's not going to be any agreement between these two technologies. So we saw genes like this, and we were kind of wondering, like, what's going on here? I think there's a lot of things that could be happening, just like you could attribute it to batch effects or other things. But what we wanted to do is kind of go back to how these technologies work and make sure we understand these correctly, so that way we can assess the problem and what might be going on. Yeah, of course.
So is it always case where you find a gene detected in Xenium but not detected in the Visium, or vice versa? Is there other cases where you find genes in the Visium but then not detected in the...
That's a great question. I don't think there were a ton of cases where it was either way. So I think this was a pretty unique case where we saw like no ApoBec3b detected expression. I think it's pretty rare that we saw that, like regardless of whether it was Xenium or Visium. But hopefully, as I talk, we'll come to realize why we're seeing no ApoBec3b expression in Visium, or why the Xenium expression has so much. Sorry, the Xenium dataset has so much expression of ApoBec3b. Cool. So now I want to kind of walk you through how does this Xenium technology actually work. So Xenium is a commercially available imaging-based spatial transcriptomics technology. Basically how it works is you have this tissue section. You're going to put your tissue slide. You're going to put your tissue section on the slide. You're going to permeabilize the cells, so that way you can send in these probes that are specifically designed to target mRNAs, to target genes of interest. So once these probes bind to the gene of interest, they will hybridize, they will ligate, and then they will amplify to become these RCA products. So it'll go through rolling circle amplification to become these products. And so once we have all these products for all of our target genes, we'll send in these probes. These probes will bind to the RCA product. We'll then take a picture of all these probes that were bound, and then we can remove these probes, and we repeat this process an n number of times, an n number of cycles. So why does that tell us where genes are? Well, let's say we have this example where in this spot, and this X and Y coordinate, the first cycle we got a green spot when we took a picture. So we washed away the probes, we sent in more. The next one we got a yellow spot. And then the third cycle we got no spot. And then the last cycle we got a red spot. So this specific barcode, the specific green, yellow, nothing, red, tells us that this is gene X present at this location. So we're able to decipher these barcodes that were previously, the probes were made to have these barcodes. That way we can say, okay, this is gene X in this spot. So that is kind of how this Xenium technology works. So with that being said, what we did is we went back to like the start of this technology. Like, how are we actually first finding these genes? And the start is sending in these probes that target genes. And so a gene is just a series of ACTs and Gs. So here, let's say we have gene X, which is a series of ACTs, Gs. We have a probe that was specifically designed to bind to a part of gene X. And so it'll be the reverse complement of this gene. And then the rest of the probe will be a sequence for our barcode, so we can decode what the barcode is. And so Xenium, or 10x Genomics, have pre-designed a lot of probe panels and that are publicly available. And so a specific probe panel that they use is this breast cancer probe panel that was used on this breast cancer data set. And so we took a look at this probe panel and asked the question, are these probes truly binding to gene X? And are they only binding to gene X? And so an really easy way to do this is using BLAT. So BLAT is just an online aligner. You can look it up right now and then put in a probe sequence like this one exactly. And you should get the same results that I get. But what it does is it takes probe sequences and it aligns these to the genome. And so here I took this probe, this ApoBec3b probe. And you can see, again, a series of ACTs and Gs designed to bind to ApoBec3b gene. And when I run BLAT, this is the result I'll get, where you can see we have our same sequence, so AAA, et cetera, and then this black line represents our sequence. You can see that this probe successfully aligns to ApoBec3b. That's great. That's exactly what we hope to see. However, we found that this exact same probe also aligns to this gene ApoBec3d. which is a problem. We have this probe that was designed to target ApoBec3b, but it's also able to target and bind to ApoBec3d. So besides the obvious issue that that is a problem in general, how does this affect our Xenium technology results? So I mentioned that probes are sent in to this target gene, they hybridize, they ligate, and they make this RCA product. We take a picture, we know that this is gene X. Well, if this probe then goes and binds to this off-target gene, we'll say gene Y, well, it's still going to bind, it's going to ligate, it's going to do this RCA amplification, and we're going to get this product. However, the problem is we have no idea how to tell the difference between these two RCA products. They will both come up to us as gene X. We cannot say that this is gene X and this is gene Y. We just, again, know that this is gene X because this probe was designed to go to gene X. It has no idea it's also going to gene Y. So now this Xenium results becomes a function of gene X plus any of its off targets. That's what the observed expression of gene X would be. And so what we did to see how many of these probes actually were affected by off-target binding is we made this tool. It's called OPT, Off-Target Probe Tracker. And this aligns probes using NUCMER to reference annotations to predict off-target binding using sequence alignment. So I did this with my friend Hayden in Dr. Salzberg lab. And then Edmund is an undergrad in our lab who's done a lot of the quantitative analysis that you have seen and will see. So this is a GitHub package. It's freely available. You can use it. I just made like a web UI, and hopefully it's really easy to use. So feel free to give it a try and let me know if there's any issues. But basically, again, all you do is you insert your probe sequences into this aligner, and it will align it and predict off targets. Now, a brief aside I want to say is you can see we have 3 reference annotations that we are using: GenCode, RefSeq, Chess. So reference annotations are basically telling us where genes are in the genome. It's saying that gene X starts here, ends here. It tells us all the inside information, where this exon starts, and et cetera. And so people have different opinions on where certain genes start and end. So that's why you'll see that there are typically multiple reference annotations. And so what we did is we used for the rest of this talk, we used the union of these results, whether we aligned to GenCode, RefSeq, or Chess, whatever the union of these results were, that's what we use in the rest of our analysis. And so we're, again, basically using all the reference annotations. And so we used OP on the breast cancer probe panel, and we found that 14 genes out of our 313 in this gene panel exhibited off-target binding to other protein coding genes based on perfect sequence homology. And so this is an upset plot showing that 11 of these genes aligned using the GenCode reference annotation, 10 with RefSeq, 9 with Chess. And you can see that GenCode had 4 unique genes, RefSeq had 1, et cetera. And so again, we took the union of these results, which is 14. So these are the genes that we found had off-target binding. And you might notice that our friend ApoBec3b was one of these genes. And so
just to clarify, for any off-target gene, would it need to be that all the probes in your target gene are homologous or identical to the off-target gene? So it wouldn't just be one, it would have to be like 4 or 5 or 6.
So it could be one probe, but in this case, it would be, these are 40 base pair probes. So this 40 base pair probe would have to have a perfect sequence match to the target or off target.
But like, how does it work? Because like each probe is just one bit in the optical barcode. So even if one bit is wrong or one of them is wrong, like off target, the next ones for that gene that you care about shouldn't be binding still, right? Or how does that?
So maybe I didn't explain it well. So the probe is binding to a gene. And so then you perform this rolling circle amplification, we get this product, and then we take a picture. So whether it's like one probe that was the problem or multiple probes, we'll still have that probe bind to that target and get that product and take a picture.
People online. This is the difference between Xenium versus Merfish. I think you were thinking Merfish, each probe has like one readout sequence. So then you end up having 40 or so probes per gene. And if any of the probes misbind, you would just get a misreadout of 2 of the bits. But Xenium, the whole probe, the whole barcode sequence is encoded in each probe. So actually there's only 2 to 8 probes per gene as compared to Merfish where there's like 50.
Thank you. Yeah.
This is like a Xenium version 1 probes, do you know if Xenium 5K are using exactly the same probes as Xenium version 1 are, or
Great question.
were those redesigned?
So we ran opt on all the Xenium public panels, and so we have those in our papers. But I think the short answer is no. I think when I ran opt on the Xenium 5K, there's still some off targets, but most of the issues were relatively gone. But I encourage you to try our tool, whatever probe sequences you have. Yeah.
So did you only look at the perfect matches? What if you allow like one mismatches, two mismatches?
Great question. Yeah. I will get to that. Cool. So if we take a look at the off targets, the predicted off targets for our gene ApoBec3b, we see that we get ApoBec3d and ApoBec3f. So these were predicted off targets for ApoBec3b. And if we can see this Visium expression of both of these genes, and you can see that these genes are a lot more expressed than ApoBec3b was in the Visium dataset. But remember, I said that because how Xenium works, ApoBec3b's expression in the Xenium dataset is a combination of all three of these genes. It's an aggregate of all three of these genes. So to correctly look at the Visium dataset and compare it to the Xenium, you need to aggregate the expression of these three genes. And so when we look at the aggregate of this expression for these genes in the Visium compared to the Xenium, you see that it starts to look a lot more similar spatially to the Xenium expression of ApoBec3b. And we can do this correlation analysis, and we can see that our correlation went from 0 to 0.18, and it has a lower RMSE. And so this is one way that we kind of evaluated if there were off targets. But hopefully you don't just take my word on it just using one technology, because Visium is just one technology. There could have been batch effects for this gene or other genes. So a way that we combat that is we use another orthogonal technology, single-cell RNA-seq. So I didn't mention that this data set also had a serial section go to single-cell RNA-seq data. And so we have serial section for this breast cancer tissue block. And if we take a look at this single-cell RNA-seq, we plotted the Xenium single cells, because remember, Xenium is single-cell resolution. And we have our single-cell from single-cell RNA-seq data in the orange here. And so you can see they're kind of separated from each other in this UMAP embedding. And so typically we would say this is from batch effects. And so we use this method called harmony, which removes the batch effects from these two data sets. And when we apply harmony and replot our data in this UMAP embedding, you can see that the cells of the single cell and the Xenium are a lot more mixed together. And so ideally, cells like in this area have very similar molecular profiles and ideally are the same or similar cell type. And so we also cluster this UMAP, and the quantitative analysis you'll see for this is using clusters rather than patch-based level expression. So to prove to you that this works pretty well is we'll go back to our friend MS4A1, and you can see that this is the Xenium expression of MS4A1 in the UMAP embedding, and it's pretty highly expressed in this little pocket. And then in the single-cell RNA-seq data, you can see this is our expression of MS4A1. It's also highly expressed in this little pocket. And our correlation analysis shows a really strong agreement between these two technologies. So then we can take a look at our friend ApoBec3b and see that the Xenium expression, it's kind of highly expressed or expressed in a couple of different areas in this Xenium UMAP. But in the single cell, again, there was no detected expression of ApoBec3b in this breast cancer data. So again, there's no agreement between these two technologies. However, if we take a look at the two predicted off targets for ApoBec3b, ApoBec3d, and 3f, we see that they are expressed, and they are kind of expressed just all over this UMAP. And if we aggregate all three of these genes on this UMAP, this is what the result that you'll get. And we can do this correlation analysis and see that it went from 0 Pearson R to a Pearson R of 0.417 and a lower RMSE. So hopefully this shows you that we can use these orthogonal technologies to kind of show or evaluate these off targets within the Xenium dataset. However, we do get some genes, like the gene TUB2B, that had no predicted off targets, but still showed a pretty big difference between the two technologies. So here, this is the Xenium expression of TUB2B. Got a really cool spatial pattern. It's pretty well expressed. And then here's the Visium expression of TUB2B, but it's pretty sparse, not very highly expressed. And so we have a pretty poor agreement between these two technologies. And so we took a look at this gene. Again, this had no predicted off targets, but when I was doing this project, I kind of manually aligned a lot of these probes just to see what was going on. And so the result I got from this is pretty interesting. So again, I'm using BLAT to align these sequences to the genome. Here's TUB2B probe, which I took from the breast panel of probes. And so if we take this probe and we align it using BLAT, we see that, okay, our sequence perfectly aligns to TUB2B. That's great. Again, that's exactly what we hope and expect. However, we found that this specific probe also aligns to the gene TUB2A, but has two mismatches in this terminal end, one at the spot 3 and one at spot 10. And so we saw this and we were like, maybe this could be the case of why we saw lower expression of TUB2B in Xenium, but we weren't sure if these probes could actually bind to these genes or not. That's when we reached out to some wet lab scientists and talked to them about kind of the chemistry and thermodynamics behind this. And they were like, yes, absolutely, this probe could still bind. Really what it comes down to is if this binding can overcome the overall binding energy it needs to bind to the target gene. So it's not about like perfect alignment. Again, even if there's a mismatch, especially on the terminal ends, which typically require less energy to bind, these probes can still bind to their target gene or these off-target genes. And if they're binding to these off-target genes, they'll ligate, they'll do rolling circle amplification, you'll get this product, and we'll still get expression for this off-target gene that would be considered for the observed gene. And so what we did is we reran opt, and we allowed for 10 base pair mismatches on either end of these 40 base pair probes. And when we do that, we found an additional 17 genes that exhibited off-target binding with imperfect sequence homology. And these are the genes here. And so you can see that one of these genes is TUB2B. And so if we take a look at the gene that was an off-target for TUB2B, well, this gene was TUB2A, as I showed you in the black. And so if we take a look at the spatial gene expression of TUB2A in the Xenium data set, you can see that it has a really similar spatial pattern to TUB2B. And if we aggregate these two genes, you can see they look very similar. And we can do this correlation analysis and get a moderate agreement between these two technologies. So in this case, yes, hopefully this answers your question that we did take a look at that, and this could be the case. So hopefully this shows you that even with mismatches, probes can still likely bind, and we get this observed gene expression of TUB2B. They may not be accurate. So we did this for single cell as well, but I'm not going to show those results for time. I'm going to move on to this gene, ADH1B. So this gene, ADH1B, had predicted off targets, but they didn't really affect the gene expression of ADH1B in the Xenium technology. So here's the expression in the Xenium. Here's the expression of ADH1B in the Visium. You can see they're pretty similar. There's some differences, but they have a pretty moderate agreement between the two technologies. But I mentioned that this gene does have an off-target. It has two off-targets, ADH1A and ADH1C. But you can see that these predicted off-targets are not really expressed. They're pretty sparse, and they're not super highly expressed. And so when we combine their expression with ADH1B in the Visium dataset to compare to the Xenium expression of ADH1B, we see that our correlation actually goes down a little, and our RMSE also goes down, but it's not really affecting our results much. And so this is to show to you that even if you have predicted off-targets, they might not always affect your observed gene expression of whatever your target gene is. So in this case, let's say you have your own Xenium experiment and you run it through your probe sequences through our tool opt, and you find that maybe it has like 10 off targets, okay? But you don't have any orthogonal technologies to check to see if those off targets are important or not, or will affect your observed expression in Xenium. Well, to answer this question and to help scientists like figure this out, so that way they're not freaking out if they find that there's off targets in their data set, we can leverage tissue-specific RNA-seq gene expression to assess how off-target binding might affect observed gene expression. And so to do this in our paper, we use probes from the HubMap program. They gave us this multi-organ panel that was designed to target kidney, lung, and heart. There are 300 targeted genes, and you can see this is the FASTA file. So this is what they'll give you is, again, just the gene, a unique identifier, and then the probe sequences used to bind to their target genes. So we took this multi-organ panel, we applied opt, allowing for 10 base pair mismatches on either end, and we found that 24 out of the 300 targeted genes had predicted off targets, which is a pretty significant amount for a 300 targeted gene panel. However, what we can do is we can utilize a tissue-specific RNA-seq data set. So I'm going to focus on the kidney here. We took the single nucleus SNARE data set from HubMap, and we can visualize the expression of the target genes as well as the off-target genes for this data. And this will give us an idea of if our off-target genes will if our off-target genes will affect the observed expression of our target genes. And so the way we visualize that is with a heat map. And so you can see here on our y-axis, we have our target genes that had off-target binding, predicted off-target binding. The first spot here is the expression of that gene. and the rest are all the predicted off targets, and then their gene expression is with the color. So you can see genes like this, ACSM2A, that has high expression in the kidney, but also you can see its off target has very high expression in the kidney. So this is a gene I would look at and be like, oh, this might be an issue. Like you want to interpret this gene with caution in your Xenium data. However, we have genes like epicam that had high expression in the kidney, but its off target didn't really have a lot of expression. Actually, it had no expression. So this is a gene, I would say, even though it had a predicted off target, it's probably fine because there's no there's no expression within the kidney of this gene. And so to show you again that this really is tissue specific is, again, this probe panel was designed for the kidney, heart, and the lung. So if we take a heart single cell RNA-seq data set and look at that same gene, ACS2MA, we can see that this gene is not expressed within the heart, and neither is its off target. So even though this gene was an issue with the kidney data set, it might not be a big of an issue with the heart data set. So again, very tissue specific problem. So the last thing I'll share is this gene HTC. And I think this gene does a really good job of showing that there really are limits to alignment-based off-target prediction. So this gene expression in the Xenium is here. It's got a really cool spatial pattern, pretty highly expressed. This is the expression in the Xenium UMAP. You can see that in the Visium data set and the single-cell RNA-seq data set, this gene is not really expressed. It's pretty sparse. And so we ran opt and found no predicted off-targets for this gene. And so I point this gene out because, again, off-targets are not like an end-all be-all. They're not going to solve all the problems and tell you all the differences between these technologies. There are a lot of other things that go into it, like batch effects, secondary structure, thermodynamics, et cetera. And so this is, again, just a tool for you to analyze your data to see if this could be the case, but it might not always be the case. So I have a couple summary key takeaway points. Hopefully I've shown you that off-target probe binding does occur in Xenium panels. We did apply this to all the publicly available Xenium panels. Feel free to take a look at our paper. That's a zip file at the end. And one thing I do want to hammer home is the reference genome annotation does really matter. I think this was a point that I didn't really understand. A lot of computational people who don't do genomics, wet lab people who don't really focus on genomics might not really understand. But I think this example does a really good job of explaining how important this is. And that reference annotations evolve over time. So here we have this probe sequence, TCF15, and you can see that this probe was designed using the GenCode version 28 version, which is a later version of GenCode. And you can see it hits this exon right here for this gene TCF15. However, in the new version of GenCode version 47, this is one of the most up-to-date, you can see this is no longer considered an exon for this gene. So this is an issue because maybe back then we had a different opinion on where this gene started and where this gene ended, but now we've determined that this is no longer an exon. So in this case, this probe probably wouldn't even bind to TCF15. It might bind to something other off-target thing or just not bind at all. So reference annotations do evolve over time. So if you are designing experiments, definitely use the most up-to-date reference annotation. Another thing I want to point out is that off-target effects likely extend beyond Xenium. So the way I can show this is a recent preprint of mine that I was looking at spatial gene expression prediction using histology images. So this is mainly what my research focuses on. But in this paper, we used this data set from a colon adenocarcinoma data set. And we have serial sections of Xenium 5K, Visium HD, and CosMx. And CosMx is another imaging-based spatial transcriptomics platform. And so you can see we have this gene, PDYN, that has little expression in both the Xenium and the Visium data, but does have a lot of expression in the CosMx data set, and it's kind of all over the place. We could maybe attribute this to off-target binding and that there are issues with these probes, but CosMx does not release their probes. So we have no idea how to evaluate and check this or to see if this is an off-target issue or something else. So for this slide is just showing that if you ever do one of these experiments, definitely try and get the probe sequences so that way you can figure out if your genes will be affected by this issue or not. And then the last thing, hopefully I showed you that it's not end all be all. If you do have off targets, I encourage you to use reference RNA-seq data sets to see if your observed gene expression in this Xenium technology will be impacted at all or not. So with that, that's all I have. Just thank you to the JeffWorks Lab and Dr. Fan for being an incredible mentor so far, and for our collaborators, Hayden and Dr. Salzberg.
All right. So I'm really excited to now introduce Dr. Jean Fan. She's an associate professor of BME in the Center of Computational Biology at Johns Hopkins. Her research team the JeffWorks lab is interested broadly in understanding the molecular and spatial contextual factors that shape cellular identity and heterogeneity. She develops new open source computational software for analyzing single cell, multi-omic, and imaging data that can be tailored and applied to diverse cancer types and biological systems. She currently also serves as an editor for PLOS Comp Bio. And the impact of Dr. Phan's work has been recognized by several awards and honors, including Forbes 30 Under 30, the Nature Research Award for Inspiring Science, the NSF Career Award, and the Presidential Early Career Award for Scientists and Engineers. So if we could give a big round of applause for Dr. Phan.
Great. Hopefully y'all on Zoom can hear us okay. So yeah, very excited to be here. I think the last time I was here, I was actually a second or third year graduate student. So now I'm on the other side. That's always very fun. Yeah, today I thought I'd talk to you all about some of the work that my lab has been doing on developing comparative spatial transcriptomic analyses. So yeah, even before I jump into things, it's always great to show you some of the faces behind the work that you're about to see. Again, yeah, we are a primarily bioinformatics research lab in the Center for Computational Biology. And when we look at a complex organ like the mammalian kidney, we're quite interested in understanding, for example, how are different cell types and cell states structurally organized in this tissue, but also how this organization and these states potentially change in the context of diseases. So for us, we do work a lot with murine models. You know, humans are very large, so it's current spatial transcriptomic technologies are still quite limited in the size of the tissue that we're able to profile. So we do a lot of work with mouse, and we can take, for example, a mouse kidney and subject it to 10x Visium HD in this case, which allows us to measure not only what genes are expressed in the tissue, but of course then also which cells express these genes. That allows us to do, all the fun dimensionality, reduction, and clustering that we saw with single cell RNA sequencing, but of course now project that all back onto the original tissue. So over the years, we've developed a number of computational methods as open source software, for example, for identifying spatially variable genes, for doing deconvolution, and so on and so forth, all of which are available as open source software with various tutorials. So I encourage you to check out our lab website if you're interested in following along. There's always demo data sets for you to tinker with, but you're also encouraged to try it out on your own data sets, of course. But today I really wanted to focus more on just one story or one series of stories on, if we have these tissue sections, let's say from a healthy versus disease setting, how do we begin making comparative spatial gene expression analyses? And in particular, I'll try to divide the talk into two parts. First, in the first part, I'll talk about if there are cell state changes that are spatial gene expression changes due to cell state changes that do not involve changes in architecture of the tissue. And in the second part, I'll talk about general cell type structural changes that are reflected in the cell type, spatial organization that, of course, then would also be reflected in the underlying spatial gene expression pattern changes as associated with those cell types. Okay, so, and in general, I think it's great to talk to a more quantitative audience. I think one question that I would like to us all to keep in the back of our head is, when making these spatial gene expression comparisons, what is an appropriate null distribution that we want to compare to in order to evaluate the statistical significance of our observed spatial gene expression trends? That's a question that will come up over and over again. Okay, so let's dive into it. So again, given these, let's say a structurally similar tissue like a kidney, which when in a disease setting like acute kidney injury, which happens when the kidney is, I guess if a person has a stroke or if we induce some kind of ischemic injury, this of course leads to a cell state change of all of the cells within the tissue, but it generally isn't enough time for there to be any major structural change. we can see this on a mesoanatomic scale also. The structure of the kidney doesn't change, but the underlying gene expression can change. So given this kind of structurally similar tissue, how do we actually make these perhaps molecular gene expression comparisons? So we developed this approach called STcompare, which enables spatial differential gene expression analysis between structurally similar tissues. This is work led by Kaylin Clifton and her undergraduate student, Vivian Zhang, who actually did a summer internship last summer with Carolyn Uhler here at the Broad. You're welcome to check out their preprint for more details. But just to walk you through how this method works, consider we have two tissues, perhaps one from healthy, one from disease. And in this case, we're showing each point is a cell, and we're highlighting one gene that has this kind of higher expression that's in the cortex in sample A, but a higher expression of the medulla in sample B. So like Caleb noted in his intro, if we just, let's say, took the bulk measurements for this particular gene, aggregated them all, and looked at the distribution of this gene across the two samples, the average gene expression is quite comparable. So on a bulk level, this gene is very similar, but of course, even visually, you can see that there is a spatial difference. So in order to, of course, statistically try to identify these spatial differences in a high throughput manner, the approach that we take is to first perform some type of spatial structural alignment. So it's kind of in order to compare apples to apples, we have to first align everything into a common coordinate framework. So in the past, we've developed an approach called STalign, which uses diffeomorphic metric mapping to register different tissue sections into a common coordinate framework based on similarities in cell density. So that's previously published work you're welcome to check out. But then after we've aligned these tissues into a common coordinate framework, this is kind of getting back to an earlier question of what is the utility of rasterization? So we rasterize these tissues so that we can, rather than making cell-cell-based comparisons, which can be actually a little bit different between health and disease, especially if there's apoptosis of different cells, but again, they're still kind of in the same locations. We create these aggregations so that we have, again, a kind of a common unit of comparison across our two tissue sections. So there's always a one-to-one matching across these two tissue sections. Because then, given these one-to-one matchings, we can do, as Caleb noted, in his intro, that we can evaluate a Pearson correlation or other types of statistics regarding how gene expression is, what is the gene expression in, let's say, sample A at a particular location, and what is that gene expression for that same gene for the second sample at the same matched location, and evaluate all matched locations. And in this case here, you can see that there's a very prominent negative correlation. And this is what we would identify, what we would call differentially spatially patterned. So because there's this negative correlation. One thing to keep in mind is we can actually, if you start with single cell resolution data, we can also do this rasterization in a cell type specific manner. So I won't actually show these results, but we have applications of this where, let's say, we only rasterize the cardiomyocytes in a heart that's been aligned. So then we're actually only comparing the gene expression in a cell type specific manner. So many different ways to do this aggregation again, at the end of the day, we're evaluating this expression at the same matched locations across two tissue sections. Okay. So of course the Pearson correlation, it gives us an R value, but a question is, how do we evaluate the statistical significance of this? Is this a significant observation, or could this negative correlation happen by chance? And I think this is particularly important because we are, at the end of the day, evaluating for a full transcriptome, 20, 30,000 genes. So this is where our question of what is an appropriate null distribution comes into play. And in general, if anyone has calculated these Pearson's correlations, you'll typically get out an R, an analytical p-value. So the analytical p-value is calculated by evaluating the Pearson correlation R against the t-distribution. So then from where your observed t-statistic falls onto the t-distribution, you can get a p-value. But one thing to keep in mind is that this analytical p-value assumes independence. It assumes every observation is independent from each other, which in this case would be assuming that all the pixels are independent from each other, which in the context of gene expression, we know is not true. And we actually leverage this not truth. The gene expression is very spatially autocorrelated in a tissue. The gene expression of one cell is often very similar to the gene expression of nearby cells because they're forming coherent tissue units, they're coordinated, and we can leverage this autocorrelation actually to identify spatially variable genes, which we've done in our meringue paper previously, and others have also done. So at the end of the day, these pixels are not actually independent. So therefore, whenever the underlying assumptions are violated, you would expect that using this analytical p-value would result in a high false positive rate and identify a lot of spurious significant correlations. So just to put this to the test, we can simulate, in this case, 100 independent random Gaussian fields in our nice little kidney simulation. So these you can think of as perhaps 100 different kidneys, the same gene, but of course, the gene is autocorrelated within each sample. So each cell has a similar expression to its neighbors, but they're all independent. They're all independent of each other. So when we compare them, we should find that they're different. So again, we can do this kind of alignment, rasterization, and evaluate at the same match pixel location. What is the correlation between all these different pairs? And in this case, we find that there is an analytical p-value of 0.49, so not significant, and that's what we would expect. But it turns out if we do it for all pairs, we do find some that you get a significant analytical p-value just by chance. So that happens by chance. But how often does this occur? Again, if we're using an alpha threshold of 0.05, you would expect this to happen if the null distribution was appropriate to happen 5% of the time. But what we find is that...
Sorry to interrupt, but just to clarify, is there some spatial autocorrelation in the simulation, or it's
correct?
There is.
Yes, yeah. Okay. So this is simulated as a Gaussian random field. So there is a degree of autocorrelation. So this is intended to proxy if we had genes that were autocorrelated, but they were independent from each other. So we would expect to see 5% false positives if the underlying null distribution was appropriate, but instead we see 50% false positives. So we're getting a lot more, we're calling a lot more things as significantly correlated, even though they're actually independent from each other. So this is just to say this can become an issue. So how do we fix this issue? The way that we decided to approach fixing this issue is by deriving a permutation-based empirical p-value, where every permutation controls for the degree of autocorrelation that's seen in the original gene expression pattern. So again, here we have our, let's say I'm trying to evaluate whether this gene is significantly correlated between sample X and Y. The way I will evaluate is now by making 100 or 1,000, however many random permutations of the gene expression pattern in X in a manner that preserves the autocorrelation initially observed. So just to dive into that a little bit more, the way we do this is, again, we can take our sample X, the pixel gene expression for a particular gene, and we shuffle it. So if I were to just stop here, that should derive actually the analytical p-value. I would approximate the analytical p-value via permutations. But we go one step further to reintroduce that autocorrelation component. So we apply a Gaussian kernel to smooth this permuted gene expression in a manner that preserves the original observed gene expression's degree of autocorrelation. So the way we do that is using this variogram estimator, which is the variance between the distances across every pairs of pixels. So it turns out, yeah, there's ways to approximate this as this function of a delta. So we can essentially evaluate a series of deltas to try to find the one that best fits the observed, again, autocorrelation structure of the original gene expression pattern. So there's a little more details in our preprint, but hopefully that gives you a sense of how we are, again, shuffling. So we're permuting, but then we're reintroducing that autocorrelation so that every permutation has the same degree of autocorrelation as the original sample. So this is what we end up getting. Again, for every x, we can permute and come up with hundreds of different permutations, and we will then evaluate each permutation's correlation with y, and that gives us a distribution. Right? So that's our permutation, permuted distribution, and then we can compare our original observed correlation to this permutation and evaluate how often do we observe a more extreme correlation, and through that approach derive this empirical p-value, which in this case is 0.54, so it's not significant. And of course, we can do this for all of our random simulations, and in this case, again, reevaluate how often under our permuted empirical p-values with this approach controlling for the degree of autocorrelation, do we find a significant correlation? And now we only get 4% of the random pairs have a significant correlation, which is within the realm of what we expect with an alpha threshold of 0.05. So yeah, so this is all just to say, again, when we don't control for autocorrelation, when we're doing this type of correlation-based assessment of spatial similarity or difference, we get a lot of false positives. If we control for that autocorrelation, we can control for that issue. Okay, it's of course a lot more fun when we look at real data. So as I mentioned, we do work a lot with kidney. This is in collaboration with Hamid Rab's group, who's the lead nephrologist at Hopkins for our transplant program. So working together, we used Xenium to profile mouse kidneys from a sham control setting. So we do a little sham surgery, and then from a acute kidney injury where we clamp the kidneys and induce ischemia, and then we perfuse, let the blood come in, sacrifice the mice. So it is a very well-established model for acute kidney injury in humans. And again, as you can see, as I mentioned, it's a very, we sacrifice the mice within 24 hours. This really isn't enough time for the tissue to completely reorganize or anything. So structurally, the tissue is still quite consistent. We still have, the cortex, the medulla, the interstitial interface. So structurally, again, the tissue is quite consistent. So we can use STcompare in this case to identify, for example, all the genes that are significantly positively correlated even after multiple testing correction according to our empirical p-value. And as you can see here, I structurally aligned the tissue. I again rasterized to a certain resolution that allows me to compare at matched locations. And yeah, so these are a few examples of genes that we find as significantly positively correlated. So I think for the nephrologists, perhaps you can appreciate a lot of these are cellular carriers and other types of cell type specific markers, because again, 24 hours after injury, we don't see any major structural rearrangement. The cell types are still where they are. So we still have, proximal tubules in the cortex, medullary tubules in the medulla, and indeed the gene expression markers associated with those cell types are still where they are. And that's what drives the high correlation And sometimes this is in a kind of compartment-specific manner. Sometimes it's cross-compartmental. Sometimes it's combinations of compartments. But generally, yeah, there is still a lot of structural contiguity driven by cell types.
Can I ask another question, which is for STcompare, I forget a little bit how it works, but is it also using gene expression to align the
great
tissues
question.
or not?
Yeah, so for STalign to do the alignment itself.
STalign, sorry. Oh, that's right.
Yeah, so in order to do the alignment to make sure that these two tissues are, in a common coordinate framework, because you can imagine if I didn't align correctly, all the downstream correlations would be off. We actually don't use gene expression. We use cell density, which is intended to, therefore not
this avoids
double dip.
Exactly. Yeah.
Exactly.
Okay,
yeah. Yeah. So we typically use if there's an image, if there's a single cell resolution measurement like MERFISH, we use the cell density. If it's something like a Xenium where it's spot-based, we actually use the histology image as the single cell proxy. Yeah, so great question. But yeah, we always try to avoid the double dipping. So we're doing our registration in a manner that's independent of the gene expression so that we can then evaluate the gene expression for similarity. Okay, yeah, but of course, so it's one thing to look at significantly positively correlated things, but it's a lot more fun to look at negatively correlated things. So again, these are the genes that we would say are spatially differentially expressed. So as you can see, sometimes they're spatially differentially expressed because they're, let's say, these are different genes. Just a few cherry-picked examples of different genes, and their kind of correlations across at matched locations across the control versus the AKI. So you can kind of see there are definitely some genes that are initially highly expressed in the cortex and then become more highly expressed in the medulla or vice versa. Other genes that are highly expressed in the interface region become highly expressed in the cortex. So when we look into what these genes are, a lot of them are related to, let's say, oxidative phosphorylation, glycolysis, fatty acid oxidation. A lot of these are non-cell type specific metabolic processes that represent, a response of how, these cells, again, independent of their cell types, are responding to the ischemic injury. So, when there's no oxygen, you're not really going to be able to use that oxygen to make ATP anymore. So I think what we're seeing is this kind of compensatory, either upregulation or downregulation of different metabolic programs, again, in response to the ischemic injury in a cell type independent manner. So that is kind of just to give you a sense of some of the things that we're able to find using these types of spatial structural alignment and spatial differential expression analysis. And again, I encourage you to check out our preprint for more details. So so then just as a summary of what I've hopefully been able to show you is that given spatial structural alignment, we can do these correlation analyses that matched rasterized pixels to identify spatially changing gene expression patterns. But if we, because of the high degree of autocorrelation of gene expression, the analytical p-value may not be appropriate. The underlying independence assumption is violated, and we can see how that leads to a high false positive rate. And therefore, approaches like STcompare try to derive an empirical p-value using permutations that control for the degree of autocorrelation to address this issue.
I have two questions. First is, for the genes that has very high correlation coefficient or very low correlation coefficient, usually what's the fraction of those also has very low empirical p-value?
Yeah, it's a great question. There definitely is a bit of a correspondence. Actually, I wonder, let's see if I can, I was gonna hide this slide. but maybe I'll unhide it. Yeah, in general, I think it's if you have a very significant R, most likely you'll have a very significant analytical and empirical p-value, right? So then it's like, where are the differences? So here are actually a few examples of differences where the analytical p-value is significant, but the empirical p-value is not. So these are the examples of genes that we filter out. And I think you can kind of see why that, again, these are autocorrelated programs that are, especially in the first example, they might be, again, autocorrelated but independent of each other. They're not necessarily medullary or cortical, but they happen to overlap at a sufficient number of locations where the analytical p-value, not accounting for the autocorrelation, thinks that there's a significant association. This is also an example where in the control case, you have a very high medullary expression, and the AKI case is maybe more of a horizontal, vertical, and a gradient that's arguably independent, but again, a high degree of autocorrelation, but these are potentially independent programs that don't necessarily overlap. So it is true that it tends to affect primarily the genes with, say, more modest R values, at least so far that we've seen in a practical setting. But actually, most genes are in this ballpark. So I think, maybe I cherry picked the ones that are a little too extreme. But the distribution of R values, most genes are in the middle.
Thank you. And the second question is, when you have multiple samples, like multiple controls and multiple disease tissues, so your strategy is, again, calculate this empirical p-value? Or?
Yeah, that's a great question. So that this is a pairwise test. So if I really did have multiple samples, practically what I would likely do is first identify the genes that are consistent within conditions. So perhaps if I had two controls, I want to find all the genes that are positively correlated, have the same spatial pattern in the controls, and all the ones that have the same pattern in the disease setting, and then only test those in a pairwise manner, perhaps. But in general, I think it's a great question of how do we leverage these statistics beyond a pairwise context.
Is there a way to like propagate, like, let's say, so you do the alignment, is there a way to propagate like the uncertainty in the alignment as to how that might affect the correlations? Because you could imagine, like, if you had like a really repetitive structure, but there's a spatial gradient and you shifted it off by a little bit, you might induce like correlations. Like, is that, and that's kind of getting a little into the weeds, but just, I'm always interested in like ways to how the preprocessing kind of propagates into the analysis.
Yes, so actually that relates to this rasterization resolution. So you can imagine, for example, when we do our alignment, we know actually the resolution of the alignment, that we know we're accurate up to 30 microns. So if there's a structure that's thinner than 30 microns, which can definitely be the case with like vasculature, then it might be aligned like this. So they're not technically on top of each other, they're kind of next to each other, but our alignment still says that's good enough. So therefore, if we rasterize at a resolution that's smaller than our alignment resolution, then these will end up being pixels that are next to each other and not overlapping. But if we use the rasterization resolution that's bigger than either the limits of our alignment resolution, then we're essentially kind of smoothing that over. So I think if there is a case where there's some kind of repetitive structure where I kind of want to smooth that out a little bit, I would choose a rasterization resolution that's bigger than that. It's a great question.
I have a question. So very cool approach. I have some thoughts like, so how should we think about, let's say, maybe generalizing similar spatial alignment of maybe like, registration on tissue samples which are slightly structurally altered? Maybe it helps, should be like assign like a tissue-wide coordinate system so that if tissue is a little bit like crumpled, the cell density changed somehow spatially, we could still maybe recover some of these interesting analyses.
Yeah, in general, it's always a great question of, do you want to do a structural alignment? So actually, maybe that's a great segue into the next part of the talk, is what happens if you don't have that structural similarity? Because you're right, especially when we start going to humans, we don't have the whole tissue section to be able to do these structural alignments a lot of cases. So for example, even in the kidney, if I profile this region in one sample and this region in another sample, there isn't a mesoscale kind of registration that I can do. But I still might want to compare spatial organizational similarities and differences. And this, I think, is even more apparent if I look at, again, other tissues. In this case, these are spleens from different individuals. Even in the context of the same individual, these are two tissue sections from the same human. And you can see there are B cell follicles, there's vasculature, but there isn't necessarily a common coordinate framework where we can register all these things together, because that level of registration that just doesn't happen at this particular scale. Sure, if we zoomed out, your whole spleen as a structure maybe is quite similar to every other spleen. I'm sure there's also these compartments. But there are definitely certain structures where, maybe an alignment and a structural comparison doesn't actually make sense. But we still want to compare cell type spatial organization changes. So this is actually where our second part of the talk, our second approach comes in. Led by Javier dos Santos Peixoto with Brendan Miller in my lab. They developed an approach called CRAWDAD, which stands for Cell Type Relationship Analysis Workflow Done Across Distances, to specifically quantify how cell types relate to each other in a manner that doesn't necessarily rely on alignment, because you're just looking at neighborhood proximity relationships. So the way CRAWDAD works, and the way that we originally really thought about the problem is that we wanted to quantify pairwise cell type neighborhood enrichments. So we wanted to ask if is cell type A near cell type B. But the reality is whether we consider two cell types as co-localized or not is often a function of the length scales that we're evaluating. So for example, if I consider myself as a whole organism, are Purkinje cells in the cerebellum and Bergman glia cells, are granule cells in my cerebellum co-localized? Arguably, yes, because they're both in my brain. But if I zoom into that cerebellum, they're actually in different layers. So you can see that in our simulation here, where, for example, is cell type A co-localized with cell type B? Well, at a very fine grain resolution, they actually do form different layers. So we might consider them to be separated. But if we zoom out enough into a larger tissue section, we might consider them to be co-localized because they are a part of the same functional tissue unit. So kind of getting back to our question of, what is an appropriate null distribution? What is the actual frame of reference that we want to compare to? This is in the case of CRAWDAD, we really do think about co-localization as a function of these different scales. So the way that we try to quantify and capture this is, the way CRAWDAD works is, given a reference cell type, in this case, let's say cell type B, we draw a neighborhood distance around all cell type Bs. So for all the cells of cell type B, we take their particular neighborhood around them of a particular distance. A lot of these distances are actually informed by what we know about physics. So for example, we know that cell-cell interactions and diffusion only really happens up to 30 microns. So we can take that as a distance. But then we create this neighborhood around all the cell type Bs and evaluate the cell type proportions of all the other cell types observed around this neighborhood of cell type B. And then in terms of the background distribution that we compare to, so that's where we incorporate this idea of a length scale. So for a series of different length scales, we create these kind of grids that then we permute the labels of the cell types within this grid. So then, we can, again, permute the labels within this grid, but we're then evaluating what is the cell type proportion of this, permuted cell types in, again, our originally defined neighborhood. But the key here is that our permutation only happens within a certain length scale. We're not permuting globally, we're permuting locally. So so given this observed cell type proportion and this kind of permuted cell type proportion, we can test the proportional, do a standard proportion z-test and evaluate are there certain cell types that are more enriched in my neighborhood than what I expect by chance based on these permutations. So that allows me to derive a z-score, but importantly, I derive a z-score at various different scales. So that's what's shown on the x-axis. I can do this evaluation of the z-score at different scales. So therefore, in this case, I'm looking at, for example, reference cell type C, which is inside this little structure. And with respect to neighbor B, we can see that they begin co-localizing at a much finer scale compared to reference A, compared to reference B, which forms this larger structure. So that's all just to say, again, B and C co-localize, and arguably A and B also co-localize, but they just co-localize at these different scales. So again, whether we consider two cell types as co-localized or not might be a function of the various scales we're evaluating. Okay, so and of course we do this for whenever we have a high throughput statistical approach, we can do all possible cell type pairs, and to just summarize all the results. We do like looking at these trend plots, but sometimes we also like to summarize all the different trends using this kind of a dot plot, where we're looking at all possible cell type pairs in combination with all other cell type pairs as the reference and neighbor. And in this case, the size of the dot relates to the scale at which it crosses the significance line. And the color refers to whether it's co-localized or separated. So we can quickly see, with one visualization on an aggregate level, what are the different trends in my tissue. Okay, so just to, again, kind of get back to this question of what is an appropriate null distribution and how does CRAWDAD perhaps also compare with other approaches. We love Gaussian random fields. So here's another Gaussian random field, two independent realizations. So these are, just like before we saw with the kidney, independent Gaussian random fields. So they're autocorrelated within each other, but independent of each other. In this case, rather than dealing with continuous variables, we're dealing with categorical variables. So we can binarize that into, let's say, a simulated cell type A and B versus a simulated cell type C and D. So again, these are autocorrelated. So you would expect cell type A to be co-localized with cell type A, but because they're complementary patterns, cell type A should be separated from cell type B. And in this case, cell type C should be separated from cell type D, and cell type C should be autocorrelated with itself, so co-localized with itself. But these two are independent of each other, so they shouldn't have any relationships. And so then when we evaluate with CRAWDAD, that's exactly what we see. If we, for example, take reference cell type A, it is indeed highly co-localized with cell type A's, and it's separated from cell type B's. They're complementary spatial patterns in our simulation, and it's independent from cell type C and D. But if we use other metrics that are very popular for looking at co-localization analysis, like Ripley's K, we find, these trends might be a little bit harder to discern. And the reason comes down, there's many different reasons, but the main one I want to highlight is, again, kind of what is our appropriate null distribution. For Ripley's, I guess for CRAWDAD, what we're doing is, again, we're evaluating a fixed neighborhood around cell type A, and we're changing the background that we're comparing against at different length scales. Whereas for Ripley's, we're always comparing to the global background density, the global proportion, and what we're changing is the numerator of the different neighborhood sizes. So in this case, compared to a global background of cell type proportions, we see that cell type A is indeed colocalized with itself, but it actually is also colocalized with cell type B here, which in our context we think is not an appropriate conclusion. Yeah, but this is, I think, more evident when we consider real data. So here is a spatial transcriptomics data of cerebellum. This is the summary dot plot from evaluating all possible cell type pairs using CRAWDAD. So I just wanted to highlight a few cases. In this case, Purkinje cells are always co-localized with Bergman glia cells, and it's always separated from oligodendrocytes, which when we visualize these cell types in the tissue, I hope you can appreciate the Bergman glia and Purkinje cells are within the Purkinje layer of the cerebellum, and the oligodendrocytes are primarily enriched in kind of the granule layer and a different layer of the cerebellum. So yeah, so if we evaluate this using CRAWDAD, we wanted to use the reference cell type as the Purkinje cells, and we find that Purkinje cells are colocalized with Purkinje cells, which is the line not highlighted here, but it's also colocalized with Bergman glia, and it's very separated from the oligodendrocytes, and most of the other cell types don't have a particular significant spatial relationship, which is, I think, consistent with what we know about the layering structure of the brain. In contrast, again, with Ripley's, I think this relationship is a little more difficult to discern. Arguably, I think this is also a correct answer. So this comes down to, again, what is the real appropriate null distribution here? You could say that the global background is appropriate if we want to ask, are Purkinje cells and oligodendrocytes, they are indeed always present, I guess, in the tissue versus outside the tissue. So, there is a realm in which you could say that at the end of the day, these are statistics. They're calculating something. I don't doubt what it's calculating. I guess I doubt whether that null background, whether the null distribution is appropriate in the context that we're evaluating here, where we do think, we actually care about where the cells are in the tissue, and there's a limited number of places the cells can be, and we don't assume that this is just a stochastic point process where cells can be anywhere. So again, I don't think the Ripley's underlying null distribution and underlying assumptions are appropriate here. And in general, I think what I like to think about is when we're evaluating cell type spatial relationships, whether I evaluate, let's say, just my tissue, or whether I evaluate, let's say, just the cerebellum here, or whether I evaluate the cerebellum with the rest of the brain. I have a slightly larger tissue section. The cell type spatial relationships I identify shouldn't change at a particular scale. So again, it shouldn't be affected by the global cell type proportions. And that's exactly what CRAWDAD is able to control for by doing these length scale-wise permutations. So actually, if you evaluate this data set and this data set, you get the same cell type colocalization patterns at the same scale, because again, that's what we're controlling for in our evaluation. So then, of course, a lot more fun to look at real data. Going back to our spleen, this was work in the HubMap consortium in collaboration with Megan Brusco from Mark Atchinson's lab. We can, again, evaluate all cell type pair relationships, and in this case, find that, even though it's hard to make structural alignments, we do see that the spleen generally falls into two major compartments with a lot of cell types colocalizing in one compartment and another set colocalizing in the other compartment. And this aligns with our known understanding of the red pulp versus white pulp organization in the spleen. But it's also, we can then, because we have so many different samples from different patients, we can begin comparing the trends. So for example, we can specifically look at pairwise cell type relationships that are very different across our samples. And in this case, just cherry picking some examples where this is a particular pairwise cell type relationship that's very different across every tissue section, even within the context of the same patient, which speaks to different sectioning, let's say just tissue sampling variation. So this is blood endothelial cells with respect to follicle B cells. So you might imagine just the vasculature is quite different depending on your section that you take. So we see sometimes it's co-localized, sometimes it's separate, sometimes it's more separate, but it's not necessarily coherent within a particular patient, which we have two tissue sections per patient highlighted here with the same colors. And this is in contrast to other cell type relationships, which can actually be quite different across patients and quite consistent within patients. So this is Ki67 proliferating cells with respect to follicle B cells. So this was actually a codex panel where we didn't have that many protein markers. So we're not actually quite sure what the Ki67 proliferating cells are, what cell type they are. But it's just interesting to observe that in this particular patient, the proliferating cells are all co-localized with the B cells. And in this patient, they're all quite far away from the B cells. So whether that has to do with, these the immune responses of these different patients at the time of the sample collection, it's hard to say with so few patients. But in general, I hope that this gives you a sense of how we can move forward with quantitatively evaluating these cell type relationships, again, across a large number of patients, and to find these potentially patient-specific trends. Okay, so with that, just to summarize the last component of the talk, that given structurally dissimilar tissues, we can still quantify and compare cell type spatial relationships, and tools like CRAWDAD can evaluate these cell type spatial relationships by comparing the cell type proportions within a neighborhood of a reference cell type compared to permutations at different length scales, which is in contrast to other methods that might compare varying neighborhood sizes to the same global background proportion. Okay. And then, I guess one minor final vignette that I think it's fun to think about is, again, these are tools that we like to think of as little Lego pieces that we combine with other tools. And one thing we're very excited about these days is, not just evaluating cell type spatial relationships across many different patients, but evaluating cell type spatial relationships or correspondences, gene expression correlation, similarities and differences, patterns, and so forth, within tissue structures, functional tissue units within one patient. So in this case, we have a nephrectomy from a human, and in the kidney, this is a zoomed in view. You can see there's many tubules. And you could say that these tubules, we can use computer vision approaches to segment out these tubules. We can analyze each tubule separately. We can consider it as its own kind of data set. And in that sense, ask, for example, what are the cell types within this tubule? Is there a particular spatial organization to those cell types in this tubule? So these are just a few examples of tubules. These are distal collecting duct tubules that are comprised of collecting duct primary cells and also CNT cells. So each point is a cell type in a tubule. But we can, of course, also do this with other tissue structures, glomeruli, and so forth. But now we can apply these same tools and say, what is the gene expression similarity across tubules? What is the cell type spatial organization? Are there certain tubules that are more intermixed? Are there certain tubules that are more segregated? And I think this becomes, in general, again, we have the computational methods to quantify these relationships, to do this in a very high throughput statistical manner and pull out interesting patterns. And I think this is, in general, a lot more exciting as we move into the context of disease, where I do think there will be certain spatial patterns to the injury itself, or perhaps one side of the tubule is more apoptotic or injured than the other. Does that side relate to how it's positioned proximally to a glomeruli or not? Those questions, again, we can begin assessing by combining these tools in combination with other computer vision segmentation or domain detection approaches. So in general, I encourage you to think about how you can use these tools, in combination with other tools to, build up interesting statistical analyses. Okay, so with that, I just want to thank again my lab in particular, students Kayla Clifton, Vivian Jiang, and Rafael dos Santos Peixoto, whose work was highlighted here. Our funding sources, our collaborators, and if you're interested in keeping in touch, my contact info is there. And happy to take any questions.
Thank you so much. And thank you to everyone on Zoom for attending.